Hello, my name is Alex Callaway, and I am the Marpan Benthic Work Package Lead here at AFBI. And I guess you could say that I am the Marpan Project Lead elect. We're in the midst of our own transition period here at the moment. And the Benthic Work Package is developing species distribution models for protected species and other species of conservation interest. We're also looking to improve seabed habitat maps for use in management of the seabed and to investigate the use of novel monitoring techniques. I have the pleasure today of introducing 3 presentations from our partners on the project. And as you can see, there are multiple contributors and collaborators to those presentations. But we have 3 main narrators, and those are Chris McGonigle from Ulster University, Rebecca Langton from Marine Science Scotland, and John Howe from the Scottish Association for Marine Science. So over to you, number 1.
Okay, it's Chris McGonigle here from Ulster University, reporting on our activities under the T2 Benthic Work Package. So this first slide is just to illustrate some of our work on non-invasive methods for monitoring in Natura 2000 sites. So here we're in Hempton's turbot bank off the north coast of Malin, exploring multi-frequency backscatter and the sedimentary composition and the influence that it's having on the acoustic response. There you can see the different frequency components there at 30 kilohertz, 95 and 300, and how these are slightly different in terms of the way the sound is interacting with the sediment. And we're starting to explore some of this in quite a nuanced way, where we can see there with these different conditional box plots how that's represented by different kind of composition in terms of material properties. And this is at a fairly advanced stage of completion now at this stage. Also, another method that we're exploring at the minute, we've had a couple of different integrations on 2 different cruises so far, and we're projected for another one this spring coming in 2021 with the ULS 500 2G robotics underwater laser scanning system, which we got this through an infrastructure grant actually outside of Marpan, but I have been able to put it to good use in this context, and it's enabled us to map with at a very fine scale structural complexity, surface complexity, and how different material properties will behave. And we're the only HEI in the UK and Ireland that have access to a system such as this at the minute. And there you can see in this plot basically a few different structural elements that are in a boulder cobble fields, and you're looking there at really an area that's about 3 meters square actually, and you have 2,048 points across that 3 meter swath, which is remarkable by comparison to most conventional mapping techniques. And if you follow that bit.ly link in the image, you can get to the model yourself and have a look. Other things that we're doing here are mapping using photogrammetry as well. So in addition to fine scale analysis of the individual images, here we're looking at combining multiple overlapping images to create a larger context that's potentially of more ecological value from the point of view of habitat mapping, and trying to find a point to integrate spatial scales here, going from fine scale remote sensing to relatively coarse scale ground validation work. And there you can see some of the products that we've produced here from the same cruise, an early one of Marpan's, between SAMS, Ulster and Imperial College actually, who came in on the outside of this. And then on the far right, you can see some of the provisional annotation work that has started here, where we're looking at identifying different morphospecies in the imagery and trying to do more quantitative work with those. From there, we've also been heavily involved in beta remote underwater video work since the start of this project, which around the time that the project was launched, we commissioned the build of an array of different baited cameras that between ourselves at Ulster, SAMS and AFBI. And during the course of the project so far, and also during COVID, we've managed to actually try and get quite good use out of these things. So there in the backscatter biodiversity shelf seas, we got 20 deployments in around the Tory Island Special Area Conservation. In November last year, we were out in the Christies in the North Channel, Middle Bank and Innistraw Rathlin area. We had 6 deployments. It was the smaller component of that cruise. And then most recently, we were out in the Sound of Jura, Loch Sunner MPA, where we got 30 deployments in a range of depths up to 350, or we had up to 350 meters of warp out in about 200 meters of water. So we had successful deployment and recovery in a pretty challenging conditions, and we got some very interesting results in this area, which is closed as a preventative measure for skate conservation, and that work synergizes very strongly with the activities of Sea Monitor. So there you can see one image representative of we're able to resolve skate quite effectively using this technique, which is very promising as a non-invasive method. Following from here, other work in the inshore or in the non-invasive monitoring, we have recently taken delivery of an inshore survey vessel, and this process was initiated around the same time as the start of the MARPAM consortium, where we were trying to basically canvass the Department for Economy to invest in ourselves to give us a platform to basically do science at sea, and we were successful, and it has taken up until now. Really, we just took delivery and have been running through sea trials, equipment integration with various partners. Here we have an inspection class ROV, and we also have full hydrographic survey capacity here. It's integrated at the minute with a Rezon 7125 and a CODA MRU and the rest. So we're ready for active survey, and we have 3 of which planned in the next few months. And these, while they're very germane to the remit of what we're doing in MARPAM, is using here freshwater lake ecosystems. We're looking at crayfish habitat and habitat use. It's going to be very interesting analog for what we've already got going. And from there, the other thing under our watch is the skate distribution modeling within T2. We have active work ongoing in that space at the minute. We're following the collation of existing data records, both in terms of the skate and also in terms of the explanatory variables that we will bring to this framework. We're assisted in this process with Sophia Loca, who's a PhD student through DERA, who's co-supervised, led by Patrick Collins at Queen's and myself at UU. And we have active work going on in this area. We're trying at the minute to recruit a postdoctoral research associate for 18 months. Owing to staffing changes, and we also have some active ship time applications in at the minute, where we would hope to do more baited camera work and synergize further with Sea Monitor in a few other sites that actually could help to connect some of the skate habitats. And that's a fairly reasonable summary of the work we have been undertaking to date.
Thanks for that, Chris. It's really interesting, and a whistle-stop tour of so many activities that are underway at the moment. And that's a really good advert for the projects, and also how they can be used to engage outside of the projects as they stand. One thing that I was hoping to see were these isopods that you found in during the last RUV survey. And some of you out there may have seen them on the MARPAM Twitter feed, but I think just the amount of information that these programs are collecting enables you to ask different questions about the system. So whilst these isopods aren't of particular interest to any of these projects, in the future you could ask questions as to whether or not these were a resident population that just took advantage of the bait being there, or if their population has been enhanced by human activities maybe, or through environmental conditions. And are they having any impact on stakeholder activities such as fishing and fish farming, which occur in the areas of interest? So whilst we won't address those questions during these programs, there's a lot of scope for future investigations utilizing the data that are generated under these 3 programs. So that's really exciting in itself. So away from these gruesome crustaceans and onto something more interesting, and that's some species distribution models from Rebecca Langton at Marine Science Scotland. Unfortunately, this recording is a little bit quiet, so if you can turn up your volume to the maximum, you'll take much more away from it. Over to you, Becky.
Over the past year, we've been working on developing preliminary distribution models for benthic species and habitats. The process for developing a species distribution model involves using data on the occurrence of a species, data on the environmental conditions where that species has been found, and building a mathematical model linking these together. The model can then be used to predict the geographic distribution of a species based on what is known about the environment. The quality of the model is therefore linked to the quality of species and environmental data used to build the model. The next step is to check the model predictions by testing it with data that was not used to build the model. This can then feed back to help improve the model. This year we've been working on preliminary distribution models for 2 benthic species and beginning to validate them. The first species we looked at was the fan mussel. This is a rare bivalve mollusk which is a priority marine feature in both Scotland and Northern Ireland. The only known remaining aggregation of fan mussel is in the Small Isles MPA, which is this MPA here, and is located up here in Scotland. There's also been one record of a fan mussel found in Northern Ireland in the early 2000s, which is this record here. But as the fan mussel is now so rare, we've had to augment the recent survey data with historic records from online databases and geo-referencing old paper records such as these on the right in order to have a reasonable data set to parameterize a distribution model. We ended up with 330 presence records for fan mussel, and these span several hundred years. We fitted a point process model to the data, which is a method that can deal with presence-only records. Variables relating to bathymetry, sediment type and oceanography were used as explanatory variables. The fitting process was undertaken 100 times, with different random subsets of the data used to train the model each time. The withheld data from each run were then used to validate the prediction. This method also allows us to map how often the model predicted areas to be suitable fan mussel habitat, as demonstrated by the map on the right. We have also been working on a distribution model for lesser sand eel habitat. Sand eel were not originally on the list of species to include as part of the MARPAN project. However, due to its importance as a prey species for many marine predators, it is a priority for several stakeholders and regulatory bodies, and the species was added to the list early this year. There are no suitable survey data for lesser sand eel in the Interreg project region, which we could have used to parameterise the model. We therefore used previous survey data from the Firth of Forth area on the east coast of Scotland. The data from grab surveys, which recorded the number of sand eels caught along with the percentage of gravel, sand and silt in the sediment. A regression model was fitted to this data to predict the relationship between sand eel density, which is the vertical axis on these graphs, and sediment composition and depth and slope. The model has been used to predict the probability of sand eel presence across the Interreg project area, and there's also data from benthic trawling surveys for the region which do record the presence of sand eel, and these are being used as an independent data set for model validation. The plan is to move on to distribution model for the habitat of 3 sea pen species next. The slender sea pen, which is shown on the left, is found throughout the project region, but the tall sea pen and phosphorescent sea pen are currently restricted to Scotland. But as you can see, for all 3 species, a lot of the records are in sea locks and very close to the coast. And previous studies and preliminary models suggest that salinity and current velocity might be important in determining the distribution of some of these species. However, many of the available layers for those variables are low resolution and don't extend to the ends of the sea locks. And as I said at the start, the quality of the environmental layers can influence the performance of the model, so this sea pen work could benefit from any layers for these variables that might be being produced by some of the other projects that cover the whole project region and extend up to the coast. And I'd just like to end by thanking you for listening, and this is my email address if anyone has any questions.
That was brilliant. Really interesting stuff, and I can agree with needing the environmental layers to come up into the shallower water so that we can improve the models. I'll show an image later on that demonstrates a very similar thing. So this was an image from one of our surveys at AFBI earlier in the year, and we were carrying out some MARPAN work during the survey from RV Christie's, the AFBI research vessel. And I thought this was a really nice image for Interreg, really, because it shows the scope of the funding area and the Marpan region, and demonstrates why we need a regional perspective. Unfortunately, it hasn't come out in the photo, but on the left-hand side of the image there was the Argyll region. We could see the islands there. And then we've got Rathlin Island directly ahead of the vessel, and off to the right, we've got Northern Ireland. And if I could have carried the panorama around further to the right, to the aft starboard quarter of the vessel, you would have seen the Republic of Ireland. So that's a really good demonstration of why we need a regional consideration of these areas, particularly for those mobile taxa that don't recognise the borders that we all hold so dear. So whilst we celebrate these shared marine areas, I'll give the platform to John Howe from SAMS and for the final presentation.
This presentation presents some of the work led by the Scottish Association for Marine Science on assessing the capabilities of an autonomous underwater vehicle for monitoring marine protected areas. Here we're going to show some of the work in collaboration with Ewan Mackenzie, a SAMS PhD student on the Bryden Centre, and also working with Chris McGonigle of the University of Ulster and particularly James Thorbourn of the University of St Andrews on assessing AUV potential for monitoring flame shells for Ewan and flap escape with James. This is a survey that was conducted in August using the AUV in Lough Carron. Now, Lough Carron is an area where there are particular beds of flame shells, Limaria hines, and the autonomous vehicle was used to monitor and map the recovery of damaged areas after the impact of a scallop dredge incident in 2017, which caused some damage. So we went up there with the autonomous vehicle and a deep trekker remotely operated vehicle, ROV, to survey the flame shell beds, the extent of the damage, and to assess the utility of these instruments in looking at flame shells and other benthic species. Here we can see two videos taken from the ROV. This is from the west beds and the east beds. So the ROV data consisted of a 180 pixel camera, externally mounted GoPro. Individual flame shell beds can be seen when the video is analysed in detail. But you'll notice there's a particular contrast in substrate where the flame shells have constructed their nests. Typical coverage is found towards the edge of the edges of consolidated reefs. Using the ROV as a drifted video provides high resolution continuous data. It's also very useful for looking at detailed guided investigations into the distribution, say, of the trawl tracks, the scallop dredge marks, or specific species ID. The ROV is deployed by a single person and is used to cover a structured area or for more detailed investigations. Sadly, in this area, the tides in the Strome Narrows in Lough Carron have quite an impact on controlling the ROV. So we went for additional surveys with another vehicle. These are two examples of data from the same area from Lough Carron flame shell beds collected by Ewan and the team, showing the differences between, on the left hand side, the trawl marks, the trawl damage over the flame shell beds, and the lower left hand panel showing nest structures, the flame shell structures. These are collected from an onboard camera on the autonomous vehicle. Now the main panel shows these grey areas. This is side scan sonar. So you can see on the side scan sonar highlighted in red the trawl damage still evident across the beds. The side scan overlays the original surveys from 2017, Moore et al. 2018, and the original damage was also recorded with drop-down video and diver surveys. So it shows that the previous damage has not recovered and is more extensive than had previously been imagined. and was seen easier for using the autonomous vehicle. So going forward, Ewan's going to analyse the stills and conduct a more detailed ROV analysis. Finally, a very recent piece of work. This is flapper skate survey, investigating the distribution of the flapper skate quite late in the season. This is work funded with James Thorbourn. And in this location, this is southwest of the Isle of Kerrera. The deep water areas are shown in blue, about 160 metres water depth. And we deployed the vehicle following a track along baited locations. And you can see the mission areas there shown by the white boxes, and we had a baited and a non-baited area. And from the baited camera sections, again using the autonomous vehicle with an onboard camera, you can see we got some quite reasonable results with conger eel, cuckoo ray, thornback rays especially in evidence. And James is continuing the analysis of the photographs, and there will be some habitat and some distribution maps coming out from that piece of work. There is a follow-up piece of work to come later in the spring, when we'll continue this on the back of the other side of Lismore.
Thanks, John. That's great, and another really good demonstration of just how much can be achieved under these programs. But I think it's really interesting to see the development of those novel technologies and just how they will inform monitoring in the future. And whilst the robotics have been around for a while, I suppose they're still quite nascent, but they're only becoming more commonplace, and hopefully we'll be able to take advantage of those in the future as well. That's great, thank you very much. And so just to end, I'd like to share this image, and this is mainly with synergy in mind. So this is a preliminary model output, and this is on the species distribution for Modiolus modiolus, which is a horse mussel, which is a species of conservation interest because it can form biogenic reef features, which are protected under European law. And as you can see, we've got areas of quite localized high probability of Modiolus occurrence, and then large areas of low probability. So this is hot off the press. It was generated by Louise Brown, our species modeler at AFBI here, and utilizing a whole bunch of oceanographic and hydrodynamic information that had been modeled by Diego Pereiro from the Marine Institute under the COMPASS program. And so I'd like to thank Diego for providing these layers to us, because they've really enhanced the predictive capability of the model as it stands. But similar to Becky, you can see we would have a preference for getting into the shallow water areas, which are unfortunately not included in some of the layers. And that's just the nature of the modeling technique. But I think that's a real good opportunity for engagement in the future. And we'll certainly be going back to COMPASS to seek more of this information that will help enhance our distribution models for the species of interest in MARPAM. And so those opportunities are there, and I think we really need to take advantage of them more often, and it's up to us to make it so. Thank you very much.